So, chapter 3, we are going to be reviewing the cell. And once again, you start off with a lot of definitions. And after the definitions, we hit our first short answer for chapter 3, and that's to describe the fluid mosaic model of cell membranes. This is very easy to do. Don't let it intimidate you. Memorize the four components. When you have short answers, you do want to memorize. It's not like multiple choice, not like matching, where you just want to be able to recognize keywords and so forth and be able to associate them. Here, you want to be able to specifically answer the question. So the four parts of the fluid mosaic model, remember what a mosaic is. It's a big picture made out of several small components, and they are glycolipids, phospholipids, proteins, and cholesterol. And what makes it fluid is the activity of the phospholipid molecules, the fact that they can twirl, that they can move side to side, and that they flex their tails. We also describe the property of the membrane that it is selectively permeable. In other words, some things can get in freely, some things are blocked or prevented. And so what can get into this particular cell? What can cross the membrane, get in or get out, either way, depending on the gradient, right? And that would be your gases like your oxygen and carbon dioxide, water, and any small hydrophobic molecule that would be able to freely pass. What can't get across? Most significantly, glucose and any charged molecule, because charged molecules, remember, don't work with lipids. They can't interact with them. And the only exception to that is water. We talked about there being diffusion, and we defined diffusion up above. And so there are three factors that affect the rate of diffusion, and that's to increase the rate of diffusion. A higher temperature would do that because it imparts more kinetic energy to molecules. Also, an increased rate of diffusion would occur with a smaller molecular size because they're able to squeeze in between things much easier. And if you had a bigger gradient, a larger gradient, where you had a huge concentration in one area or region or volume of a solute versus another volume. And so the bigger that difference is, the more molecules that are going to move from the area of high to low concentration. Why don't cells usually burst? Well, we have to go back to our idea about osmotic pressures and in the hypotonic state. And so this had to do with our isotonicity. If you recall, we had the definition of a tonicity above, and we had three different states. We could be hypertonic, where the extracellular fluid concentration is greater than the concentration of the intracellular fluid, so water will move out and the cells will shrink. We have isotonic, where everything stays the same. And then we have hypotonic, where the concentration is lower outside the cell in the extracellular fluid than it is inside the cell in the intracellular fluid. So water is going to move into the cell. It'll keep getting bigger and bigger and bigger due to this osmotic pressure. Why doesn't the cell just blow up? Well, there's going to be a pressure that develops against the push of water into the cell, and this is called hydrostatic pressure. So as water builds up inside the cell, it builds its own pressure. And eventually the pressure inside the cell from all that water entering it will counter the pressure of the osmosis that's pushing the water into the cell. And eventually they'll meet up with each other and equalize, and the cell shouldn't burst. This can be overwhelmed in certain circumstances, like people that drink too much water, overhydrate themselves. So this can be overwhelmed in certain unusual situations. We then got into the concepts of passive transport, facilitated diffusion, and active transport. And you should just understand passive transport employs both simple diffusion, which is the movement of molecules across the cell membrane, those that can diffuse across that we discussed earlier, gases, lipid molecules, water, and so forth, that'll move across a membrane and don't need anything to help them move. They diffuse freely. But then there's the molecules that can't, but then there are proteins in the cell membrane that allow these molecules to move across the membrane according to their concentration gradient. And so that would be facilitated diffusion. It's still a passive transport because there's no energy required. That's the key thing. That's why it's called passive. Anytime you see that word, that means no ATP is used. But remember, passive transport is two types of diffusion: simple diffusion and facilitated diffusion. And all facilitated diffusion is simple diffusion that requires a protein to occur. So it's facilitated, right? It needs help in order for the diffusion to occur. Then we got into the notion of active transport, and simply when you hear the word active, you know that ATP, energy, is needed to be supplied for the process to occur, for the gradient to be applied. So because what we're going to do here is we're going to push a molecule against its concentration gradient. And that's not something that happens naturally. We need to supply energy in order to make that happen. So over these rather small processes that move a few molecules at a time, perhaps, we needed to talk about processes that move large molecules, large amounts of molecules at a time. And that would be your exocytosis and endocytosis. And exocytosis just involves a formation of a vesicle inside your cytoplasm of a cell, and then that vesicle will migrate to the membrane, fuse with the membrane, and release the contents. That's exocytosis. And we'll be talking about that when we talk about the release of neurotransmitters or hormones or secretions from glands. Endocytosis, a little more complex, not necessarily in the process because it's basically the reverse of exocytosis. A vesicle will form in the membrane, will capture whatever material is surrounding it, and then will enter the cytoplasm, eventually to interact with the lysosome, probably. But there are three ways that this can be enacted. And a vesicle can begin forming on the surface of a cell through what's called receptor-mediated endocytosis. So an external molecule, like a hormone or a neurotransmitter, can bind to a receptor that will enable the formation of this vesicle. It will invaginate or enfold, pinch off, and then enter the cytoplasm. And whatever was attached to that receptor has now entered the cell, and we can process. The second type of endocytosis is known as cell-eating, or more appropriately as phagocytosis. This is often employed by immune system cells that want to digest a microbe, a germ, and begin eating it. You form the vesicle based on contact with a microbe. The vesicle forms, encapsulates the microbe, and it intersects with the lysosome to destroy it. And finally, the last process is a random process known as penocytosis, or also known as cell drinking. And since it's random, you just get this random formation of vesicles off the cell membrane. One neat thing about endocytosis and exocytosis is that they result in a renewal of the cell membrane, a constant renewal, because these processes are always going on the surface of your cell. We then moved on into the cytoplasm. We had 50% of the cytoplasm is liquid or cytosol, and the other 50% is organelles. We're going to focus on the organelles now, and we begin with the cytoskeleton that's actually composed of three different components. That's composed of microtubules that are made up of the protein tubulin. These are the largest or thickest of the cytoskeletal elements. They're so important, they're so involved in positioning the organelles within the cell. The organelles just don't float around, they're actually held in anchors by these microtubules. They also will are responsible for forming flagella, cilia, elements called centrioles, and microtubules function to position things because they're shaped like baby beads. They're very short, and so you can attach these units of tubulin to one another and increase the length of microtubules, causing things to elongate, or you can remove them and you can shrink the size of the microtubule and cause things to shrink. The second type of cytoskeletal element is the microfilament. It's made up of proteins called actin, and it is the thinnest of the cytoskeletal elements. These often are found near the cell membrane in the cytoplasm, and they have little anchors that extend out to the cell membrane, and they're going to be very important in the division of the cytoplasm. And so when these microfilaments begin to shrink in size like a lasso, like they start to shrink, then it will pull on the cell membrane in that region, and you can shrink them to such a small amount that it will divide the cell's cytoplasm in half and split it into two separate cells. The other type of cytoskeletal element that I don't list here is the intermediate filament, and it's just kind of a go-between. It serves as a support for the framework of the microtubules. Centrosomes, centrioles are used for mitosis. Centrosome is the region of the cell where the centrioles are located. Think of them as big old rootstock, like for a redwood tree or something. And from here, we're going to grow microtubules for all the things that are important, especially in mitosis. Cilia are short projections from the cell membrane, hair-like projections. They number in the thousands and are found in the reproductive tract and the respiratory tract and so forth. Flagella are usually, you only find one, and it's a very long projection from the cell membrane made up of microtubules, and it is associated with sperm cells. Ribosomes are found only in the cytoplasm, at least in their active state. They can be attached to the rough endoplasmic reticulum, or they can be free, and their job is where proteins are manufactured. The rough ER has ribosomes attached to its surface. As you see here, the definition, we're going to see that when the ribosomes make the proteins, they'll enter the channels of the rough endoplasmic reticulum, and they'll be modified. Eventually, those proteins that have been modified will be sent through a vesicle transport to the Golgi body. The Golgi body then tags and ships them to their correct locations in the cell. Smooth ER is primarily where lipids are assembled. The Golgi apparatus is the UPS center of the cell. It's like we just talked about. From the Golgi apparatus, we can produce lysosomes. These are the vesicles full of about 40 toxic enzymes. They clean up the cell. They're your general debris collectors and get rid of all nasty little structures and organelles that don't need to be there. Peroxisomes are primarily tasked with detoxifying effects associated with alcohol consumption. Proteasomes are these little vesicle structures that are responsible for getting rid of old proteins that have degraded. Your mitochondria is our next major organelle. It's the power plant, or where most ATP in the cell is formed. And finally, our last organelle of importance is the nucleus. This is a double lipid bilayer. Remember, that's the hallmark here is for the nuclear envelope, is it's double lipid bilayer, not just a single one like surrounds the cell. Through it, though, we do have nuclear pores. There has to be a way for molecules to pass both into the nucleus and out of, but it is an incredibly selective process. Within the nucleus, we have chromosomes. We'll talk a little bit more about those in chapter 4, and a darkened region called the nucleolus. It is a place where RNA is stored as well as other critical proteins. Now we get into chapter 4, and here we talk a little bit more about DNA. Possible short answer, be able to name the three major differences. DNA is double-stranded, RNA is single-stranded. DNA has a 5-carbon deoxyribose sugar, but RNA is called ribose. RNA has uracil as a base, DNA has thymine as the base. So they both have guanine, cytosine, and adenine, but instead of thymine, RNA employs uracil. Their similarities are, of course, structural. They both possess sugar phosphate backbones. They both have the nitrogenous bases that we just talked about, and they both have the 5-carbon sugars, just different forms of them. From DNA, we transcribe into the molecule called RNA, ribonucleic acid, and there are 3 types of RNA produced from DNA, and that is messenger RNA, which is your instruction manual for producing a protein. You'll produce ribosomal RNA, which is your place where the protein is assembled, and transfer RNA that supplies the amino acids for the growing protein. Most important thing, make sure you understand that in the nucleus, transcription of DNA results in RNA. You're just taking 4 bases of DNA, transcribing it into the 4 bases of RNA. Then that RNA molecule is going to exit from the nucleus and will enter the cytoplasm where translation will occur, and you'll have the RNA to protein. Chromosomes, defined as just a DNA molecule plus associated protein. And it's incredible that a chromosome is only about a third DNA molecule, but about 2/3 protein. Humans do have 46 chromosomes. Centromere, normally in the normal state of the cell, while it's not undergoing division, a chromosome is single-stranded. If there's a single component to it. It's a double-stranded molecule. It's a double-stranded helix. I don't want to get you confused there. But there's only a single component to it. During a portion of the cell cycle called the S phase, it will be duplicated. Then those 2 components of the chromosome will be joined at what's called the centromere. The 2 components are called chromatids, right? And genes are just short stretches of DNA. Not all parts of a DNA molecule code for a protein, just small stretches of it. Okay, you should know that the genetic code is what is used to convert the 4 bases of RNA into 20 amino acids. You should also know that all words in the RNA, they're known as a codon, and each of these codons is 3 letters long. The part of the cell life that is occurring when there is no cell division is called interphase, and there are 3 parts to the interphase. So the first stage of interphase is called G1 or GAP1. This is where you're living life good, you're making all your proteins, lipids, carbs, everything's great. And then once you have made enough of those, the cell will get a signal that it's ready to go into division. And so to start that process, you're going to duplicate your single chromosome. And then once that's done, once all the 46 chromosomes are duplicated, you're going to go into G2 and produce proteins that are necessary for mitosis. Mitosis is only division of the nucleus, though. It is not division of the cytoplasm. That's cytokinesis. Know your stages in mitosis. This is great for matching. I'll probably list the stages, and you need to match the stages to what events are occurring in them. Know that chromosome number does not change during mitosis. If you start with 46 chromosomes with 1 cell, when you come out with 2 cells at the end of mitosis, both those will still have 46 chromosomes. Okay, chapter 5, tissues. We proceed on. Notice there's no red additions here. That's for you to add to. You should define all those terms in the first line. Go back to your slide, list 5 functions of epithelia. Go back to that and do that, so fill that out. You should be able to determine the epithelial type when I give you a word description. For instance, let's say I'm going to give you a word description such as the following. Basement membrane. Know that basement membrane of all epithelial tissue. So already you know I'm talking about epithelial tissue. Then I say the next describing term is a single layer. So now you know it's a simple epithelial tissue. The next describing term is that it's squarish. And then the final describing term is found in kidney tubules. And hopefully from those 4 describing terms, and I'll say select the tissue type from your choices below. And you'd select simple cuboidal epithelial. So that's just the way to start thinking about, okay, so I'm not going to get a picture, I'm going to get a word description, and how do I figure out what Dr. Elliott is describing in this particular description. All right, so once again, basement, some particular characteristics here of epithelial tissue. Has basement membrane, there's very little material between the cells, it has no blood vessels. What are the surfaces called? The surface that faces the lining, right, the skin, inside your body for tubes and things that faces the opening is called the apical surface. The one that is attached to the rest of the body is the basal surface, and then you have the two side surfaces known as lateral surfaces. You should look up your three types of cell junctions: tight junction, desmosomes, and gap junctions. Tight junctions are found in between cells. Typically, the primary example we gave was the brain, forms the blood-brain barrier, and it's like a needle and thread. You literally sew up the membranes together so that no water can pass between them. So the only way things can pass this barrier is through the cell. So the cell has to have specific transporters in order for things to pass it. And that's a great thing for the brain. The brain doesn't really have much of an immune system associated with it, so it needs a barrier to prevent free entry of things that can hurt it, like pathogens, like microbes and so forth, and then other potentially toxic compounds. The second type of junction, the desmosome, is really something that's you can call a spot weld. It's like a big chunk of solder you throw onto a cell, two cells that are next to each other, and it just holds them together. So these are found everywhere. This is your general type of membrane of a cell junction that's going to hold these things together. And then finally, the gap junction directly connects to adjacent cytoplasms together. It's like a little tunnel through the cell membrane so that two different cells with the gap junctions between them act as a single cell. And we talked about these with the heart. The heart couldn't beat as a single unit if it didn't have gap junctions between the cardiac muscle cells that exist. All right, then we got into the idea that there are two different types of glands because all glands are made up of epithelial tissue. There are endocrine glands. Endocrine glands are those that secrete their products into the surrounding fluid, which eventually then migrates into the blood vessels and gets transported through the entire body. Most common example is hormones. Hormones have a very widespread effect throughout the body because they're carried by the blood. The other type, exocrine glands, have ducts. They secrete their products into the ducts, and the ducts transport the products onto a surface, like the skin. Sweat glands, for instance, right? And so sweat, oil, milk, earwax, all of these are examples of exocrine functions. So they have a very local effect because they're only going to have an effect on the area of the skin or wherever they happen to be released at. That's the only area they're going to affect. We can classify glands based on their secretion modes. If they're merocrine glands, they release their products through exocytosis into the duct and onto the free surface. Turns out the second type is exactly the same way, called apocrine glands. But because the type of secretion product is different, we're going to keep them separate. But the way they release their products is still through exocytosis. And then there's holocrine glands that release their products by degradation of cells, right? The cell actually breaks down and releases its product. And so there has to be a lot of mitosis going on around there to keep producing these cells. And the types of cells we talked about here were sebaceous glands that secrete oil that coats hair. We then got into connective tissue. You should look up your at the most five functions of connective tissue. Blast, create the matrix. Cytes, maintain the matrix. Clast, break down the matrix. Adipocytes are fat cells. The extracellular matrix is really what connective tissue is. It's what gives the body structure and form, and its hallmark is ground substance. So extracellular matrix is the ground substance whose consistency will determine the function of the connective tissue. And the three types of protein fibers are collagen, perhaps the most abundant in the human body, reticular protein, and elastin protein that provides for elasticity. Ground substance will also have hyaluronic acid for energy, also proteoglycans that determine the water consistency of the ground substance. And consistency of ground substance can range from fairly semi-fluid to very hard. Types of cells that produce all connective tissue, whether you're an adult or whether you are a growing embryo, are the mesenchyme cells. So then we got to the various types of connective tissue. We had loose connective tissue. There are two subtypes, areolar and reticular. These two subtypes of loose connective tissue both possess fibroblasts for creating the extracellular matrix of the loose connective tissue. It's a semi-fluid or syrupy ground substance. Areolar has both types of fibers. It has elastin and collagen fibers. It's going to be found just beneath the stratified squamous layers of your skin. It has a lot of white space in it, so there's not a lot of density to the extracellular matrix here. The reticular type of loose connective tissue hallmark is it has these dense reticular fibers that form a netting to filter fluids like the lymph fluid that carries potentially dangerous microbes in it. This netting is found in lymph nodes and in the liver. Dense connective tissue comes in two subtypes as well, irregular and regular. There's less ground substance associated with these, but it is a much firmer ground substance than found with loose connective tissue. Of course, also possesses fibroblasts, fibrocytes, and so forth. Dense irregular is where the collagen, very little, no elastin in dense regular or irregular connective tissue. The dense irregular connective tissue then has collagen that is in random orientations with each other. This is great for forming a protective capsule around organs. The dense regular connective tissue has the collagen in parallel, and so this confers strength that you would need with tendons that connect bone to muscle and ligaments that connect bone to bone. Our next type of connective tissue is cartilage. There are three types you should know: hyaline, fibrocartilage, elastic cartilage. The cells that produce cartilage are chondroblasts. The cells that maintain it are chondrocytes. You should know the locations of the three types of cartilage. Hyaline cartilage is found in joints. Fibrocartilage is found in the intervertebral disc, and elastic cartilage is found in your external earlobe. Now, you won't have any pictures to identify. Everything that I'm going to ask you to identify will be written, as the example I gave you above for the epithelial tissue. Bone has a very hardened ground substance. It's hardened due to the presence of calcium salts called hydroxyapatite. So just get that in your head right now. Those are synonyms, calcium and hydroxyapatite. The cells that produce the bone matrix are osteoblasts. The cells that maintain the bone matrix that I didn't list are osteocytes. The cells that break down bone matrix when we need calcium are osteoclast. And a stem cell that leads to the direction of any of these of osteoblast is osteogenic cells. The last type of connective tissue is blood. Why is it a connective tissue? Well, first, it's made by bone marrow, so it's made from tissues in the bone, and it is a transporter. That's one of the functions of connective tissue. It really has no protein fibers in it, but when it begins to clot, it does have an extracellular matrix that can form with these different proteins connecting together. It also has red blood cells, white blood cells, and platelets. Finally, yay, the last line is know your three categories of internal membranes. So that excludes the cutaneous membrane. So we have the mucous membranes, the serous membranes, and the synovial membranes. You should know any special names, characteristics, and components. So for mucous membranes, we have a couple different types of epithelial tissue that can form the outer layer. The inner layer is a areolar CT, connective tissue layer, and it's known as the lamina propria. For serous membranes, they have an areolar CT inner layer, but their outer layer is only simple squamous and is known as mesothelia. And finally, for the other internal membrane, it's called the synovial membrane. It has no epithelial, and it is only a mixed bag of connective tissue. and it's found in joints. Mucous membranes are only found, as far as locations are concerned, mucous membranes are only found surrounding tubes that open to the outside. Whereas serous membranes are only found lining areas or cavities that do not open to the outside, and synovial membranes line joints. So yes, you should know locations as well. Hope this helped. Good luck, and we'll see you for Unit 2 and Chapter 6 in the next videos.